Snehalata, and for the invitation. I will like to talk to you regarding my career path and how immature I was when I started my career. Almost no understanding. I came here as a physics student in this country, as a master's student in physics, and then I changed to statistics and did a lot in genetics. So that's a brief introduction, but I will go through the details of it. So now I am in University of Louisville, Kentucky, Department of Bioinformatics and Biostatistics, and I get to do too many things now. So life is not easy, but I do enjoy it quite a bit. Okay. Now, when I came, I just finished my bachelor's, and I was doing master's in University of Calcutta, and my boyfriend then we got married. He came to this country as a PhD student in statistics. And then for a while I thought I will complete my degree and I come here, but it so felt that we needed to be together. So I applied in Michigan State as a physics student, and I got admission and a financial assistantship, so I came there. And then I was doing physics masters, but I saw there are people, very brilliant ones from across the world, and they were doing their second, third postdoctoralship. Then I thought that I cannot continue like this. I got to get a real job sooner than that, so I wanted to change the career. So in the meantime, he was in statistics, my husband, and then he got a job after finishing 3 years PhD. He got a job in University of Georgia. Then I decided I will move there anyway. I have to change. So by looking at him, and in between I started taking probability courses, and one of the probability courses within the physics program that I have taken, it was taught by an Indian professor, Professor Ramamurthy in Michigan State. It was so very entertaining and interesting. I said, I can handle probability. That's not a problem. Then my husband coaxed me, how about going through statistics, maybe? I said, okay, very related, so I had no idea really what I'm getting into. But I said, okay, anyway, I have to change to either computer science or probability, something like that. So when we came to Michigan's University of Georgia, I changed to statistics, and I got a master's degree in applied statistics. I did not dare to go to theoretical statistics because I didn't know anything about statistics, but I survived. Then I got a little more encouraged that I can handle it. It seems very easy to me, so I decided to continue with PhD. But I wanted to be very different from my husband, because I decided that if I have to go to the job market, then I better specialize in something which is absolutely different from my husband. So there was a genetics professor, adjunct in statistics. He started his career as a statistician, then moved to genetics. So he had joint appointment in statistics and genetics. Basically, he was doing theoretical population biology with probability, my favorite subject, and lots of stochastic processes. So I started doing research in theoretical population biology with stochastic process in it. So that's dynamics of cytonuclear disequilibrium. It's evolution, theoretical population biology. So that was my PhD thesis on. But then after graduation, not after graduation, even before graduation, I was thinking now it's the job securing time. Again, there is a problem that we may have to get separated, coming from all the way from India, living with so many people. Before marriage, I did not even stay without my family. I was always within the family, living with my mom and dad, and staying apart was not a choice. I wanted to make sure that I don't get separated from my husband. So what I did, I started looking locally what is available. University of Georgia is in Athens, Georgia, and the closest big city is Atlanta. So I started looking in there. And there is Emory University. They had a postdoctoral position opening, but it's in a different subject. In statistics, it was in biostatistics department, but it was in infectious disease modeling. I thought about it. I thought, okay, I know basic statistics. I know probability, so okay, I know genetics quite a bit. So how difficult can it be? So I took this job, 2 years postdoctoral position. It went fine. I mean, I published my thesis in between. It was a 2 years postdoctoral position. And then I did some work. The group says that it was a path breaking in their group, so they got energized. I got a publication pretty quickly in working there, so that bought me some amount of flexibility. So sometimes I didn't have to go to work regularly. I could work from home. Why I'm saying this, I had to go, but it was flexi hour because that's when I decided to have my child. I wanted to make sure everything falls into place. I did not want to be a woman researcher without any child. So my daughter came there. It was end of second year almost when she came, and I took just a 6 weeks break, and in between I was applying for work. So when I went for the job interview, the baby still hasn't come. I mean, I was fully pregnant when I went for job interview. I'm lucky. There was only one. That's the only job application I did in Georgia State University, again, downtown Atlanta. I was very lucky to be able to convince them that a fully pregnant woman can do this job. So some amount of luck is needed for you to succeed. So I got the job, but it was compromised because that department at that time did not have PhD program. I knew I'm really going to ruin my career to some extent. But after joining, right away I started working on how can I bring the PhD program as soon as possible. So as an assistant professor, I really worked maybe triple times with a baby, tried to make the PhD program happen somehow. And as I knew a lot of genetics, there was an initiative at the university to develop bioinformatics program. So I worked really hard with the chair of biology and computer science so that we can develop this program. And while I was there, it happened. So bioinformatics program was there, and I did manage to have one PhD student in that program. And I graduated 10 master's theses. So I made sure that I'm not hurt. In future, if I have to go somewhere, that I'm not hurting my research career. So I worked as hard as possible to be marketable and to feel good about myself. But that department, we all worked hard, and research was a priority. So I got my first NSF while staying in that job, and so I got promoted fairly quickly to an associate professor. And I was there from 2002 to 2005 as an associate professor. And then I got a joint appointment in Department of Biology. as well. So, but then again, another phase of life, my daughter was growing up. She used to go to school. Then the school choices came into play. So we were living near Atlanta, but she was going to school. But Atlanta is, traffic-wise, it's terrible, just like any other big city. So I was driving like crazy. We were living sort of in between in Gwinnett County, but it used to take me like an hour to commute to work. So, and my daughter used to go to school in Athens, Georgia. Because of that, my husband used to take her there. For 12 years we did this, but then we thought it's not working. It's just too much. Daughter has to live near the school in order for her to do all the social activities and all. So there came the time we decided we have to move. But by then we need, my husband was a professor, so we need one professor's position, one associate professor's position, which is highly unlikely to get. So we wanted to look for some place where it is building up. That's when you get these positions. So University of Louisville at that time, they had an ad, so I just tick mark, I check Amstat News, and then I made that a note. But in between I went, how amazing is this? I went to MBI, Ohio, and I gave a talk there. And in the social hour we were talking, I was talking to Ray Carroll, Texas A&M. I was just telling him, he's a good friend of ours, I was just telling him that, you know, we're thinking about moving. And there comes the chair of University of Louisville. He heard it. So he approached me. You guys are looking for jobs? Then I connected. Then he said, why don't you come for an interview? I said, okay, fine. Just contact me. I will go there. And it worked out. So what happened, I went for the interview. They were very happy. They wanted to hire. Then I brought in the fact that, you know, with other faculty members, that I have a husband who is a professor. I'm telling you, it works much better if the women get a job and then asks for the men's spouse to go. That's the way to go. If you go there with your husband as a spouse, you will never get your respect. That's the unfortunate reality. But here, I got the job. My husband tagged along. Both of us were well respected. And then we basically built the department, worked really hard, built the department so well. Now we are sending our PhD students to Harvard, Google, every place. So we basically built the department there. But for anything and everything, some luck factor is there. So there is no unique recipe. But in order to get the respect, one thing is for sure, to make your research valuable, publish no matter what. No matter how difficult your life story is, publish your work. That's something you can control. And then don't get disappointed. Write grants. If it gets rejected, fine. You know, just submit. But of course, seriousness is needed. And what I have seen, you may not be really happy with wherever you are. Very few of us are 100% happy with where we are. But try to get the best out of the situation. Wherever you are, it has its strengths and weaknesses. But concentrate on the strength. Then you will get somewhere. Of course, if when I was looking for the job first time, my advisor got me a position with Elizabeth Thompson in University of Washington. She's an authority of statistical genetics. Maybe my profile would have looked even better. But I have to compromise thinking. It's a complex problem of optimization. You have to, family is there, child rearing is there. Everything, when it falls to, on your shoulder, that how to optimize it, it's the decision you have to take, and then don't regret. Let things happen the way it is. Then I got, it was a new situation. I got my tenure. First promotion was very easy for me, but when I went there, I was hired as a tenured associate professor because, again, I had the grant, and also I was an attractive candidate there. Then I waited there just to feel the water there, so applied a little late for my professorship in 2010. I wanted to make sure there is no force over there that can deny my application. So I waited for a while. So 2010, I applied for a professor's position, got it. Then 2010, right after that, I applied for university scholars position. It's a prestigious position. At the same time, it gives you extra money. So at this stage, I really don't care much for, some respect. I do, but if somebody gives me more money, I'm really happy to take it. So, I got that, and then this position is every 3 years. It has to be renewed, so it's like evaluation after every 3 years. So it's quite a demanding position. There are strings attached to it, what you have to do, grant and publication, this, that. So when I reapplied in 2013, I got promoted to Distinguished University Scholar. That's the highest you can do at your institution. So I'm happy for 3 years. 16, I will see how it goes. So anyway, that's my life story. And then 2012, we got new chair and all. After he has come, he gave me the graduate director's job. It's fine. I enjoy the student interaction, but at times it is really involved. And then I have this distinguished research position and then the graduate director. It's really a lot of work, but so far it's going all right. Okay, so now I'll concentrate on my research talk. So as I said, I have different research areas: genomics, proteomics, statistical data analysis for genomics, proteomics, metabolomics, lipidomics, all the omics, big data stuff, and then survival analysis, and infectious disease modeling. You saw that. So today's talk, I will concentrate on genomic data. It doesn't have to be from microarray data. It could be next generation sequencing data as well. But I don't know whether you guys are familiar with this data. So gene expression data is where you are seeing the expression of thousands of genes, maybe 40,000 at the same time. And since 1995, it came in 1995, I think, the first data. Scientists were trying to see whether the genes behave differently under different experimental conditions or case control study. Gene expressions change for over the time in different tissues, on case versus control, whether they change, but mostly it was done like every gene was observed. It's on a chip. All the genes you were studying on a chip. However, one gene separately, they were seeing every gene, how are they changing independently. But people have realized, especially biologists, they have realized that physical and functional landscape of a biological system, can be revealed by network. It's not that each individual gene or protein, they're acting on their own. It cannot happen. Everything is there together. All the genes are, even if not all the genes, but most of the genes or proteins, they are acting together. So it could be realized as networks. So here specifically we'll talk about genomic networks. I do work on protein-protein interaction networks as well. What has been done so far is most of the work has been done just to construct the reverse engineer one such network under one condition. So it's a static network, just one condition. You, out of those gene expression data with expression of different genes, you construct a network. But this is a network. How does it work? So the nodes are the genes and they are connected. The connection is constructed with so these are edges of the network, and these are like some associations. The strength of the association between the two makes them connected. Okay? So there is a threshold associated with it. And these associations, these are association-based networks. So these are some scores that we calculate from the gene expressions for this gene, that gene. So whether you do it individually, just combinatorially from this to this or not, that's not the point. I'm just saying this score can be calculated. It could be correlation, it could be partial correlation, whatever it is. But from that, we can draw a big network. This is a static network under one biological condition. We will come back to this network later. But see, this is the data now. But since 2012, people have been observing, very few people, not a lot of scientists, but there has been talk. Trey Idacker's group in San Diego, they are doing a lot in this area also. They are trying to see whether these regulatory networks, they are different in different conditions. So is it static or it is very dynamic, the very condition specific? What we have seen, this is example 2. Believe it or not, it involves the same genes but in different conditions. It looks very different. The other time you have seen all of them are connected. Here in another condition you have 5 different modules. This is another experimental condition on the same genes. So it is very dynamic. The network structure, the topology of the network is different under different experimental conditions, or if, under different tissues, it's very different. So we say that the differential network analysis has to be done. We want to see that how these networks are changing across different experimental conditions. We want to study the networks. We want to find the difference between the topology of the networks under 2 experimental conditions. What's the statistical underpinning of that? So that's what we are going to study. Visually, you can see there are some differences, but is that difference statistically significant? That's what we are trying to assess whether. So before that, we will talk about that how we have constructed the network to begin with. And then we will talk about how we are going to see the changes in the network. So how we are going to do that? A pair of strongly connected gene in one. Okay, before that I want to give 2 counterexamples that a pair of strongly connected gene in one condition may not remain connected in another condition. Those graphs that I have shown you, it is a good example of that's what happens. Many genes, they were not there, they were not connected, so it got lost from the network. On the other hand, a pair of weakly connected genes under one condition may be very strongly connected in the other experimental situation. Okay, so let's first discuss how we did the construction of static network to begin with, we have done the static network construction, and one of our very best student, of course, we lost him to Google. He was a computer geek, basically. He could do computing really amazingly. He went to Johns Hopkins for a postdoc, but even then he liked computer a lot more than statistics, unfortunately, so we lost him, but he still does statistical research there. So why did we get involved? Why did they even need statisticians or mathematicians for this kind of problem? Because initially they tried to do a lot of knockout experiment biologists, and then they did Yeast-2 hybrid study. I'm not going into the details of this. APMS, affinity purified mass spectrometry techniques, and all these experimental procedures enables the biologists to construct network. However, there's a seminal paper by Bader et al. There they have said the reproducibility of those experimental networks, they're terrible. So then they came, these are very time-consuming experiments and very expensive experiments, but it doesn't result reproducible results. So they thought, there was a thought process going on, maybe because we have too many genes and proteins, maybe the computational models, if the mathematicians or statisticians, they come up with something just through the data, maybe they will set up their experiments that way. So that may be a feasible model. So that's how it happened. So we found out the construction of static network. I will talk to you even more about that. But then comes the question, how do we study the differential network analysis? So we have two such static networks under condition 1 and condition 2. So how do we figure out the differences, really, in a very informed manner, not just by looking at the graphs, but in a very mathematically or statistically handleable way? So the 3 questions we have come up with: whether the modular structure of the two networks constructed with the same genes are different under 2 experimental conditions. So we will find the modular structures of the 2 networks and see whether they change under 2 conditions. Next is whether the connectivity of a particular set of interesting genes. So these interesting genes you can find out from just individual studies, or in some pathway, some genes are there you are really interested in, you want to study whether their connectivity gets changed or not when there are other genes along with it. It is very different whether these genes are differently expressed under 2 different experimental conditions. That's not what it is. Whether their expression in the presence of all others are, so the connectivity of these genes, whether that's changing under 2 experimental conditions. So that's the problem here. Number 3 is whether the connectivity of a single gene has changed between the 2 networks. So there are 3 sets of things. This is a single gene, whether that gene has changed the networks. This is a cluster of genes or some number of genes, whether they have changed their expression values in the presence of others between the 2 networks. So these 2 are kind of related, 2 and 3. But first one is very different. Okay. So the building blocks of our statistical test are the sets of scores that measures the strength of gene-gene association or correlation. So that's the initial stage of network construction. So as I said before, that the association can be measured with correlation, partial correlation measure, mutual information criteria, posterior probabilities, which measures the association of one gene to the other. So that's the score. We will focus on association based on partial least square regression. I mean, why partial least square regression? We were trying many other things in this paper, And we have seen that partial least square regression was giving us, in multiple simulation studies, this was giving us better results in terms of sensitivity, specificity, and all. So we used that. And then we came up with this because in 2001, I wrote a paper in gene-gene interaction, just like that. It was a very, what should I say? I just thought about it, I wrote it, I learned partial least square, and then I thought, okay, let me see how it works in this context. But this is a good paper to read because it tells the strength of partial least square regression. Partial least square regression actually is used in chemometry, not that much in statistics community. So how is the data? So the data is gene expressions. The raw data is you have the multiple genes under experimental conditions, the expression values. So it is usually normalized and then log transformed. That's how it is. And you have the data metrics. It's of sample size n and p many genes. Usually this p is way larger than the sample size. That's how it is. 40,000 genes under 10, replication size 10. If you get 10, you are lucky. Usually 3, 4 in smaller institutions. So very, very ill-posed problems. But anyway, so correlation association is measured by the simple correlation between the xi, gene xi and xk is given by this. Partial correlation, it's given by this quantity where wik's are the entries of inverse correlation matrix. So we are talking about inversing the correlation matrix. And right away, if n is smaller than p, there's a problem. It's not invertible. The matrix is not invertible. In that case, one can use Schaeffer and Strimmer. They have shown that if you take the Moore-Penrose matrix, but Moore-Penrose matrix, the solutions is unstable, so you have to do it multiple times. So they say it's bagging, bootstrap sort of thing, so to get some estimate of this. And then some people have came up with a shrinkage estimator of the covariance matrix, correlation matrix, and that's how they have found these things. There's a huge literature on statisticians. They really like the Gaussian graphical model because anytime the Gauss comes, we are happy because it has all the nice properties in it. So, but partial least square that we have used, it's not as nice, but it's very cute. It gives nice results. How does partial least square work? So basically, we are trying to find the association between ith gene with all others. So a basic regression model would have been xi equals beta 1, let's say I minus 1, all the other genes beta 2 times x2, whatever it is, all the genes. So you have too many beta components to estimate, but you have very small sample size, so how it doesn't work that way. How much time? Couple of minutes? Oh, okay. So anyway, so we have done this. Okay, I will skip the details of it. So this is the score we have come up with, which constructs the network. The slide will be given, so you can read some. But then, in order to test the overall difference between the networks or differential network, the module of network, this is very important. What is a module of network? A collection of genes in the module of, let's say, F, will be called a module. It has to have certain cardinality, let's say M. Given two genes, F1 and F2 in F, they are connected by a path of genes for some k greater than equals 2. So it has to be connected. There is some threshold that they have to be, the score that you calculate, it has to be greater than some number, and you have to have a certain number of genes in that network in order for it to be module. So the tuning parameter here is epsilon, how strong the association has to be, and how many genes has to be there in order for a part of the network to be called a module. So that's the idea. So that is user selectable. And then, so we have come up with a test statistic. Wow, I don't have time. Time flies. With this n statistics here, it says it is like proportion of non-overlap statistics. The idea over here is if this quantity is, it lies between 0 and 1, 0 indicates there's identical modular structure in the two networks, and 1 indicates that the module in the two networks have nothing in common. Anything in between, there is some relationship in there. So now this particular quantity, it's very hard to find out what is the null distribution of this. It's asymptotically, it's quite a hard problem, especially in the context of partial least square. So because nothing much has been done on this is fairly new to statistics community. So what we did, what everybody does, permutation-based test. Okay, the permutation-based test, the idea is that suppose under the null hypothesis, you know that there shouldn't be any difference between the two networks. Then if you change the levels, exchange the levels, you ignore the levels of these gene expressions, I don't care which network it comes from, you have a big matrix where everything is jumbled up. So you just generate the data. Let's say first 10, you say it's network 1. Next 10, you call network 2. And construct that statistic. Do it randomly many a times and see what you got from your data. How odd a sample is that? Okay, that's the idea of how you calculate the p-value. So n pi is the value of the statistics under pieth permutation. So keep doing it. So p-value is estimated as proportion of times the n pi, the statistic, the pieth permutation statistic is greater than equals what you got from your observed data. So that's how you calculate the p-value. Similarly, you do construct the test statistics for the group of genes, and then you estimate the p-value, again with permutation test. Do the same thing for a single gene, construct the test statistic, and then permute it to find the p-value. So those three conditions that I said. Then we constructed the simulation. We did some simulation. The simulation scheme we generated the data from a very, very different kind of network. This Michaelis-Menten and Hill kinetics equations. We have generated one such genomic network and tried to see how does it work. Our method, how does it work with this? It had 50 genes, 50 samples like that. So network A looked like this, 5 modules. Network B looked like this, and this is drawn by Cytoscape. If you want to do anything with network, these are powerful tools. And of course, it depends. These are tuning parameters, and this is m equals 5, the minimum modular size module. We call something to be a module if it exceeds 5 genes or so. And then the sensitivity analysis is done with epsilon, and then 1,000 permutation tests. So we got this result. It was you have seen on the graphical basis that it looks different, and our test is saying that's the thing. And then we did a real data analysis with the graphs that we have seen. It's on a mouse model with lean and heavy mice. I'm not going into the details of the experiment. However, I showed you this picture. This was the lean reconstructed networks for lean mice using our method, and this is the heavy mice. So it's very plausible. This is experimentally also it is makes sense because for lean mice, you can control with gene-targeted therapy. Let's say you target this heavy, this hub gene. These are called hub gene. It's connected to many, many genes. Then you are okay. But if you are talking about heavy mice, you cannot do anything. Okay. So I guess I will, and then some biological analysis with these, some of the genes that we found. It's very highly probable. It gives us the trust that what we are doing does make sense. This is a picture of two different pathways of this network. And we have the R package. It is available here for constructing differential networks. And the conclusion is that differential network analysis provides formal statistical tests to explore how network structure containing multiple gene changes between two conditions. This is often done in conjunction with a differential gene expression analysis that can be used as a filtering. Sometimes you have to do this filtering because network you cannot handle with too many genes. It becomes quite cumbersome. But there's work to do. We want to do the asymptotic null distribution and the multiple hypothesis correction. How to do that, it's not done yet. It's an open problem. And then want to acknowledge the grants, of course, and thank you for your attention.